Hello everyone, welcome to Visionary First Step YouTube channel. Today I am going to explain the topic Plant Kingdom. Whittaker in 1969 proposed 5-kingdom system of classification where he explained detailed about Monera, Protista, Mycota, Plantae and Animalia. Now Monera is about bacteria, Protista is about unicellular eukaryotes, Mycota is about fungus, Plantae related to plants and Animalia is related to classification of animals. In the second topic, biological classification, we already discussed the detailed characteristics of Monera, Protista and Mycota. So in this topic today I am going to explain plant kingdom, and in the next video we are going to discuss about the animal kingdom. So here understanding of the plant kingdom has changed over time. What is the reason for this? Initially, taking general characters, they placed all the organisms together except animals like fungi, bacteria and Protista and cyanobacteria, all are placed in plant kingdom. Now when they got to know that the composition of cell wall of bacteria, Protista and Mycota is different. We also know that bacteria has peptidoglycan as a cell wall, fungi has chitin as its cell wall, and Protista, because it includes all the unicellular eukaryotic organisms, you cannot say particular cell wall, but some of them has cell wall, some of them do not. And cyanobacteria, initially it is considered as blue-green algae because of its pigmentation and color, and they are autotrophic. But now they got to know that cyanobacteria are the group of bacteria, they have peptidoglycan as their cell wall. Now all these organisms, very important, fungi, Monera, Protista and cyanobacteria are excluded. This is very important, excluded from the Plantae. Okay. Now once these organisms are excluded, the classification of plants are like Algae, which is the most primitive plants. In what sense we can say primitive plants? In the sense there is no true root, stem and leaves. The body of the plant cannot be differentiable. It is just like a mass of cell. So algae are the most primitive one. That doesn't mean all algal forms are having primitive structure. There also certain classification is there. You can observe advanced type also. So here plant body is called thallus. Okay. In the Bryophytes, see the difference here. Algae are aquatic. Either they are found in fresh water or they are found in the marine water. And there is no land adaptation. But when it comes to Bryophytes, they are commonly known as amphibians of the plant kingdom. And there is a reason for that. They can survive on the land, but they require compulsory water for their sexual reproduction. Okay, so they are named as amphibians of the plant kingdom. Here we can see root-like structure called rhizoids which help the plant to adhere to the substratum. Now they are slightly advanced than algae. Now coming to pteridophytes. Now these are the first land plants. Pteridophytes are the first land plants. Land plant means they should have some adaptation for the land condition. Plant should adhere to the surface of the soil with the help of root. That means here body is well differentiated. You can see true root, stem and leaves in the case of pteridophytes. And in Gymnosperms, they are much more advanced than Pteridophytes. Actual vascular system, that is xylem and phloem, develops in Pteridophytes, and that will get advanced in Gymnosperms, and most advanced type is observed in Angiosperms. Now the gymno name is because of naked seed. That means seeds produced in the plant is not covered by the fruit. So the name Gymnosperms. And highly advanced one is Angiosperm. As we all know, angio means seeds are covered by fruit, and these are the flowering plants, very commonly observed in every place of the earth. Angiosperms. Okay, now they are the highly advanced one. Now coming to the next part. Now this is the classification system, helped in classification of plants. The first one is artificial system of classification. When we heard this name, artificial system of classification, because we are all from Zen generation. Artificial means AI, not like that. Artificial system name appears like it is most advanced one, but it is the most primitive type of classification proposed by Carolus Linnaeus. In detail, we will study in the next slide. And another type is natural system of classification proposed by Bentham and Hooker. And the recent one, not completely organized one, but in the process of completion of classification, that is phylogenetic system of classification. The recent classification was proposed by Engler and Prandtl. Let us go in detail of each classification one by one. Now the first one is artificial system of classification. Very interesting. Carolus Linnaeus proposed it. In artificial system of classification, they have taken only gross morphological character and floral character, few floral character like number of androecium for the classification. And they have given equal weightage to vegetative character and floral character while they are classifying the plants. Now, of course, this is the basic for classification. Somebody, how they get to know, classify the organism, this is how they make trial and errors. And here, there are some limitations for this classification. The reason they have taken vegetative character and floral character with equal weightage made this classification to classify some of the unrelated plants in a particular taxa. At the same time, related plants are placed in faraway taxa. This created a problem. So, this artificial system of classification has some limitations. And when they are given the equal weightage to vegetative character and floral character, vegetative character is more often is affected by the change in the environmental condition. Now let us take one example for this. There is two habitat, one species of plant. One is growing in an area where soil is having very less amount of water, in the sense we can call it as xerophytic condition. Another place where soil is having sufficient water for the growth of the plant. Let us consider it as mesophytic condition. Now the same species, one species growing in two different areas. Now when there is a good amount of water, leaves are developed fully. But when it is grown in the area where water is less in the soil, at that time what happens? Size of the leaf will reduces. See, same species, but there is a morphological difference. So this is what we are discussing. When vegetative character and floral characters are given equal importance, vegetative characters are more often affected by change in the environmental conditions. So with these limitations, this classification cannot be holds good. So by knowing the importance of that, Bentham and Hooker, these two scientists, they proposed the natural system of classification. Till today, if you study taxonomy, we are using natural system of classification for the classification of plants. Okay, so what makes this natural system of classification till today we can use it? What are the characters? Natural system of classification also considered morphological character, but along with that it also considered internal structures like ultrastructure. You know, ultrastructure means the minute characters even present inside the cell can be observed with the help of electron microscope, and those characters are also taken for the classification. And anatomy is taken. Anatomy means internal character. Anatomy and embryology is also taken. See here, ultrastructure, anatomy and embryology. Embryology in the sense, when we look at the different embryos of different species, they are not exactly same, but related species may have similar embryos. So which will help to add the character to classify the plants. Okay, next one, phytochemistry. Phyto means plants, chemistry means chemicals. So the plants have certain amount of chemicals like alkaloids, phenols, even the nucleic acids, proteins, these are all chemicals. The similarity and dissimilarity helps them to classify the plants in a particular taxa. Okay, so this is the character taken by the natural system of classification for the classification of plants. Now coming to the next one, that is phylogenetic system of classification. So this is the recent classification proposed by Engler and Prandtl, and based on evolutionary relationship. This is important. What is evolutionary relationship? So if we trace back our ancestors, we'll have some common characters. The advanced one will have more character, the primitive one will have less character, but there will be a connection. Okay, it is not totally different. So the evolutionary relationship is one of the important character to be studied under the classification. So phylogenetic system of classification fulfills that. And this is one limitation in natural system of classification. So by knowing the limitation of this, Engler and Prandtl, they proposed the phylogenetic system of classification. Okay, so for this they take fossil evidences. I hope you know what is fossil. We all studied in our lower classes. During the volcanic eruption or during the landslide or any catastrophic event, when it occurs, the living organisms get trapped, and for millions of years their body remains intact inside that structure. And if you cut open that rock or sedimentary rock, inside that you observe this trapped organism that we consider it as fossils. Okay. Now, moving on to the next. So this is important again. Whatever we studied for the classification so far is not sufficient. So other source help to resolve the difficulties in classification. Even though we have external character, internal characters, anatomy, ultrastructure, embryology, phytochemistry, phylogeny, having all this character, but there is some requirement still to get the better classification in plants. So these sources like numerical taxonomy, cytotaxonomy and chemotaxonomy help us to overcome those problems in classification. So let us study one by one. What is numerical taxonomy? This is very interesting actually. Present days, having information is very easy because we all have computers. So there is no need for a person to study taxonomy, go and visit particular what we can say herbarium or something else where the plants are stored to study its character. In the click of a button, you will get all the characters. How is this possible? They made it possible by taking all the characters. They have given codes and numbers to this. So any person from any part of the world can access all the characters in a very short interval of time. So this helps in classification. So that is about the numerical taxonomy. Here we use the computer. Okay. Then what is cytotaxonomy? Cytotaxonomy, cyto means something related to cell. So inside the cell we know it has nucleus. Inside the nucleus DNA is present. During the cell cycle this DNA undergo condensation and form a chromosomes. Now we are focusing on chromosomes. Chromosome, number, structure and behavior, very important. Chromosome number, this chromosome number may not be same in all the organisms. Chromosome number will vary and its structure. All the chromosomes are of not similar sized. Some chromosomes are larger, some chromosomes are smaller. Okay, then their behavior, mainly at the time of crossing over. These things we will study later in Cell the Unit of Life and Cell Cycle, Cell Division, and in second year Principles of Inheritance and Variation. There are so many topics linked to this. Anyway, in cytotaxonomy they consider chromosome number, structure and behavior. Now coming to chemotaxonomy, already we are aware of this term chemo. Chemo means chemicals. Chemotaxonomy means what? Here, chemical constituents. Chemical constituents means just like just now we discussed, proteins, nucleic acids, alkaloids, phenols. So these are all taken for the classification. Taxonomy means grouping of organisms. Okay. Now this is about the other source help to resolve the difficulties in classification. Now the next part is algae. We are directly moving on to the classification of plants now. Very interesting part and very, very important for your competitive exam. Okay. So we'll go in deep and study different examples under algae. First we will discuss the general characteristics of algae. What is general character? Suppose somebody asks who is Harsha sir, how you will explain? By looking at my morphology, right? Similarly, in general character, their morphological characters are considered. Along with that, some reproductive character is also considered here. Okay, let us go by one. So algae are chlorophyll bearing organisms. So the chlorophyll will vary here. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, like that it varies. But algae are chlorophyll bearing organisms. And they are simple, this is important, and thalloid. Why the name simple? Because body cannot be differentiable like true root, stem and leaves. So the body form is very simple. And the name thalloid is taken because it cannot be differentiated into true root, stem and leaves. Okay. Now autotrophic and largely aquatic. Autotrophic means the organism have ability to synthesize their own food by using the sunlight. Then only we can call it as autotrophic. All the algal members are autotrophic. They are not depending on any other organisms as a source of their food. Give an example in your textbook. There may be possibilities some algae are parasitic, but whatever the example given in your textbook, they are all autotrophic. Largely aquatic. So here aquatic in the sense some of them may be freshwater forms, some of them may be marine water forms. Freshwater forms may in the sense they may be found in lakes, or it may be found in pond, well or streams or in rivers. So these are the aquatic freshwater habitat. Marine water means in sea, okay, salt water. They occur in variety of the other habitats. Now coming to habitat, where they live actually. Do we commonly observe algae in all the seasons? No. In summer seasons, we rarely observe algae. Wherever there is a continuous moist environment, there we can observe the algae. Otherwise, algae, they disappear. That means we observe algae on the moist soil, moist rock, and sometimes on the moist bark of the tree. Or, see, this is very interesting. Algae sometimes observed on the body of the animals. So how can we expect the growth of algae on the body of the animal? The best example is sloth bear. Sloth bear is very slow moving organism and its body is suitable for the growth of algae. Okay. And one more interesting character, algae sometimes associate with the fungus, and that we call it as lichens. Here fungus is heterotrophic, algae is autotrophic, and they together forms the lichens. So these are the different habitats we observe in algae. Now coming to the form and size of the algae, this is highly variable. There is a reason to quote this entire sentence in a color. This question is very important for your competitive exam. See, some examples are given here. The size ranging from colonial forms like volvox, filamentous forms like eulothrix and spirogyra, and few marine forms like kelps, they form massive plant bodies. See, whenever we think of algae, we think just it is like a thallus. But there is a change in its body. Some of them are colonial, that means many unicellular like organisms, they are together and form a colony. Filamentous in the sense, cells are stacked one over the other and forming a ribbon like structure, that is called filamentous, observed in eulothrix and spirogyra. And apart from this, some algae are just like giant plants. Do you believe algae can grow up to 100 meters? Yes. Kelp, they are very massive, growing in marine water, reach up to 100 meters. Look at the diversity of their body form. Okay, so this is the body form character. Now coming to this, these are some pictures I added here so that you can visualize it. Just now I explained about Volvox. This is the Ulothrix filamentous, this is colonial, this is also filamentous, and this is the massive body of kelp. Okay. Now, reproduction in algae, how they reproduce. Generally, when we take the primitive organisms, we consider vegetative mode of reproduction, asexual mode of reproduction, and sexual mode of reproduction. Sexual mode of reproduction is the advanced one. Vegetative mode of reproduction is very common, that is by fragmentation. This thallus, by some mechanical injury, undergo fragments, and those two fragments grow into new plants. It's a very simple form of reproduction. So the number of fragments may be n number. All the fragments grow into new plants. Okay. Then in the case of sexual reproduction, this is very important. Remember this. Again, we study the sexual mode of reproduction in the classification of algae, that is Chlorophyceae, Phaeophyceae and Rhodophyceae. So make sure you understand each and everything about this sexual mode of reproduction right now. Now there are three types: isogamous, anisogamous and oogamous. Let us try to understand each and every word here. Iso means what? Similar. Gamous is related to gametes. Gametes when they are formed during the time of sexual reproduction. So in isogamous condition, produced gametes are of similar sized. Very important. Now here again there is one condition. These gametes may be motile or sometimes non-motile, but they are similar sized. Then only they are classified under isogamous condition. So two specific examples are given, very important for the NEET examination. Gametes flagellated, similar sized in the case of Ulothrix. Gametes non-flagellated and similar sized in the case of Spirogyra. These two examples are very, very important. Now coming to anisogamous condition, again aniso means different sized, gamous in the sense gametes. So the gametes produced, we cannot say male and female gametes here, plus and minus we can take. They are of dissimilar sized, and here also it is not given, they may be motile or non-motile. The example given is Eudorina. Again, this example is very, very specific and important. Let me tell you why. See, in Chlorophyceae, Phaeophyceae and Rhodophyceae, the classification of algae, they have given some examples. But in those examples, Eudorina is not added. So remember, again, remember this, very important, Eudorina belongs to Chlorophycean members, and the unique characteristics of Eudorina is they produce anisogamous gametes. Okay. Then the next one is oogamous type. So what is oogamous type? See, oogamous in the sense, here two gametes are produced. One is small and motile, another one is large and non-motile. Okay, so fusion of one large and one non-motile, that is static female gamete, and they are smaller one is male gamete and the larger one is female, observed in Volvox and Fucus. Again, these examples are very, very important for exam. Okay, that is oogamous type of sexual mode of reproduction. Okay, next part of the topic is economic importance of algae. So to study anything, we should know what is its importance. Okay, and then only we will have some seriousness towards the study of those organisms. Here, there are some certain interesting economic importances. We found algae in very less number and they only appear during the time of rainy season. So this is all we studied. But just look at the numbers here. At least half of the total carbon dioxide fixation is carried out by the algae. Really interesting, right? Half of the total carbon dioxide fixation in the sense photosynthesis. During the time of photosynthesis, carbon dioxide gets fixed and it forms the glucose in the presence of sunlight, and that is done by the algae, half of the total carbon dioxide produced. Now, being photosynthetic, they increases the level of dissolved oxygen in the immediate environment. Now, what is this dissolved oxygen? See, in the atmosphere, we can breathe easily because of the presence of oxygen. At the same time, in the aquatic habitat, whether it is a pond or a lake or any type of aquatic ecosystem, ocean, it requires oxygen for the aquatic organisms. So that oxygen is released by the algae. Okay, so it increases the level of dissolved oxygen in water. Actually, the level of oxygen, dissolved oxygen in the aquatic environment decides the more number of aquatic animals. Okay, then it can act as a primary producer. So again, in lower classes we studied some of the things like primary producer, secondary producer, tertiary producer, like this. Primary consumer, secondary consumer, and tertiary consumer, like this. A producer in the sense, by using the sunlight, it has ability to produce the food. Okay, so it acts as a primary producer. And these examples are very important. Porphyra, Laminaria, and Sargassum. These three species and some other 70 different species are marine algae, and they are used as a source of food. Okay, there are many examples in which algae, having all the nutrients required for growth of the organism, can be used as a source of food. Now, certain marine brown algae and red algae produces large amount of hydrocolloids. Let us see this new word, hydrocolloids. See, hydrocolloids are the type of material which have the ability to hold the water. In our childhood days, we sometimes use this type of hydrocolloids. Some small ball-like structures are in some packets, and when we put those ball-like structures in the water, they swell, and they are used in keeping some plants hydrated and placing in their nearby roots. Okay, so which are those? Actually, algin, very important, by brown algae, carrageen by red algae. Here, actually, cell wall is made up of cellulose, and surrounding that, in the case of brown algae, surrounding the cellulosic cell wall, they have a coat of algin. In the case of red algae, surrounding the cellulosic cell wall, they have carrageen around the cell wall. So these algin and carrageen acting as a hydrocolloids. Now question comes, why they have algin and carrageen? Just look at their habitat. These organisms are marine. Usually they grow in the shallow water region. Shallow water region means the depth of the water is very less. So whenever the low tide condition is there, these plants are easily exposed to sunlight. So at that time, plant may die due to dehydration. So in order to avoid that, on the surface of its cell wall, it has algin in brown algae, carrageen in red algae, that protects the plant from not undergoing dehydration. But it can be economically usable as a hydrocolloids. Okay, then next one is agar. Very important here. See, all these I have mentioned for NEET examination, very important statements. What is agar? Very new technical term it is. Agar is a substance commonly used in nutrient medium preparation. Now, what is nutrient medium preparation? Now, my question is, can we grow fungus in the lab? Answer is yes. Can we grow bacterium in the lab? The answer is yes. Can we grow plant in the lab? The answer is yes. Now, the question is, can we grow the plant in the lab without soil? The answer is yes. The condition is, to grow any organism, you require the essential nutrients of that particular organism. Once you know the essential nutrients, then according to that you prepare the nutrient medium. Now, usually when we prepare nutrient medium, it is in the liquid form. Now, suppose I am going to grow a carrot, so that I have taken a small piece of carrot having totipotency. It means total potential of the cell to grow into a new plant. I placed it in the broth medium. At that time, the whole cell will sunk down. So, what can we do? We will make that medium solidified. To make the medium solidify, we require agar. Now, from where we can extract this agar? Agar is extracted from two red algae, that is Gracilaria and Gelidium. These two are the example of red algae, Gracilaria and Gelidium, from which agar is extracted. And one more important character of agar is, it is not degraded by any of the enzymes. So, when the plant or any bacterium or the fungus in the medium starts to grow, they cannot digest the agar. So, the solid medium remains the same, but nutrients will be absorbed. Okay, so that is the importance of agar. Now, here they have given one more importance, that is it can be, the agar can be used in the preparation of ice creams and jellies. Nowadays you can see jelly chocolates, so those are made up of agar. Now, next one, chlorella. Very, very important, chlorella. Again, chlorella having all the type of nutrients can be used as a source of food. Even human beings can consume this chlorella. So, space travelers, to avoid the weightage of their food, they use the tablets prepared from the chlorella. So, it can act as a very rich nutrient source of food for the human beings. Okay, so these are the economic importance of algae. See, this is one question appeared in NEET 2022. Hydrocolloid carrageen is obtained from? Option 1, Phaeophyceae only. Phaeophyceae only, Chlorophyceae and Phaeophyceae, Phaeophyceae and Rhodophyceae, and Rhodophyceae only. Now tell me what may be the answer. Here they have specifically mentioned hydrocolloid carrageen. They have not taken algin and carrageen, only carrageen is taken. Then the answer would be Rhodophyceae only, because algin is present in brown algae, that is Phaeophyceae, and carrageen is present in red algae, that is Rhodophyceae. Very simple question. If you just observe the pattern of question, they have changed from 2022 to 25. Now the questions are more descriptive type. Here they found out one technique where they can kill the time of the students while they are writing the examination. So the questions are very elaborate. Long sentences are taken for the preparation or framing the questions. Okay, so let us see those questions in the later part. Okay. So next one, Chlorophyceae. As I said earlier, algae are further classified into Chlorophyceae, Phaeophyceae and Rhodophyceae. Let us take general introduction for these three classes. Chlorophyceae, green algae, Phaeophyceae, red algae and Phaeophyceae, brown algae, Rhodophyceae, red algae. Now in Chlorophyceae, there are some examples given. First we will take the example and then we will go to the general characteristics. Some of the example I already shown, Volvox, Eulothrix, Spirogyra. Then in addition to that we have Chlorella, Eudorina and Cara. So these are some examples for Chlorophycean members. Now coming to its common name, commonly known as green algae. Now the question comes, why they are called green algae? Because they appear green in color. Why they appear green in color? Because they have the pigment that is chlorophyll A and B. Chlorophyll A and B is the dominant pigment, so they appear green in color. Okay. Now pigments are localized in definite chloroplast. So this is also important. Pigments are not freely present in the cytoplasm of the cell, but they are localized in definite chloroplast. Okay. Now chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped. So there are 5 different types of chloroplast are given here. Shape of the chloroplast is not same in all the species, it differs. Now discoid shape means like this, almost discoid shape. Then plate-like shape, reticulate somewhat like this, then cup-shaped like this, spiral shaped, okay, or ribbon shaped. Ribbon shaped means like this, ribbon shaped, filamentous like. So these are the different shapes. Okay. Now most of the members have one or more storage bodies. Now tell me why these storage bodies are present. Very common. See, plants, they undertake photosynthesis. At the time of photosynthesis, they produce large amount of glucose. Now some of the glucose is immediately used for the cellular respiration. What about the remaining glucose? That has to be stored, because all the time is not favorable condition. On set of unfavorable condition, plants, they use the stored food material. In the same way, algae, they store the food, but not in the form of starch what we observe in higher plants, but here in a special type of structures called pyrenoids. What is this pyrenoid actually? The centrally one protein is present, surrounding that starch is observed. Okay, here I have given actually pyrenoid contains protein beside the starch. This is very important statement, protein beside the starch. That means a central dense protein matrix and often surrounded by a sheath of starch. That is the pyrenoid. Please make sure what is pyrenoid so that you can remember it, because this difference of storage materials in Chlorophyceae, Phaeophyceae and Rhodophyceae is very important character. Okay. Next, some algae may store food in the form of oil droplets. This is the general character. Then green algae usually have rigid cell wall, and that is made up of cellulose and pectose. Why this character is important again? Because in Phaeophyceae, surface of the cellulosic wall you observe algin, and in Rhodophyceae, surface of the cellulosic wall has carrageen. Okay, that is not observed. So here it is cellulose and pectose. Now the next part is about the vegetative reproduction. See, this is interesting. In all the organisms, vegetative reproduction is by fragmentation. There is nothing to worry about this. But coming to asexual reproduction is by flagellated zoospores and produced in zoosporangia. Zoosporangia is the specialized structure to bear the zoospores. Now, why zoospores? Because they are motile. They have a flagella. That is why it is called zoospores. Now, one thing will make sure the dominant plant body of the algae is gametophyte. Please remember this. And one more thing, gametophyte is always haploid. I will repeat once again. The dominant plant body of the algae is gametophyte and gametophyte is always haploid. Now, when the gametophytic plant body by the asexual mode of reproduction, when it produces the zoospores by mitosis, these zoospores are haploid. N number of chromosomes are present inside the zoosporangia. Zoosporangia is nothing but what? One of the cells on the thallus get enlarged with the sufficient reserve food material and nucleus inside that undergo many times division and becomes more number of nucleus, and around that nucleus again cell wall formation takes place. Now inside one vegetative cell so many zoosporangia are present. When the cells burst open, these zoosporangia are released to the environment and they grow into a new plants. So this is the asexual mode of reproduction. Okay. Now the sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous and oogamous. This part of the topic we discussed in detail previously. Isogamous means gametes are of similar sized. Anisogamous means dissimilar type of gametes. Oogamous means one gamete is smaller and motile, another gamete is larger and non-motile or static. Okay, so fusion of gametes takes place with the help of water because they are completely dependent on water for sexual mode of reproduction. Okay, so these are some of the commonly found algal examples, that is Chlamydomonas, Volvox, Eulothrix, Spirogyra and Chara. In the next slide, I have shown all the pictures of these organisms. Please observe. Here, Chlamydomonas, they are unicellular organisms. Here you can see their flagella also. And Volvox, this is the colonial form. Eulothrix, filamentous form. Here you can see cells are stacked one over the other to form a ribbon like structure. Okay, then this is chlorella, again unicellular organism. What is the importance of chlorella? Chlorella, it is used as a source of food by space travelers. Spirogyra, just look at its structure. Here chloroplast is spiral in structure, so the name Spirogyra. This is Chara. Chara, somewhat it appears like a higher organism. Okay, somewhat leaf-like structure is observed in Chara. We commonly observe Chara pond. They are mosquito repellent organisms. Wherever in the pond, if the Chara is present, the mosquito cannot lay its egg apart from its your syllabus. It is just additional information. Okay. Then this is Eudorina. Where we studied this example of Eudorina, do you remember? Anisogamous type of sexual mode of reproduction. Example is Eudorina. Okay. So the next one is Phaeophyceae. Just observe the name here. Pheo means what? Brown color. brown algae. So the name brown algae. When it comes to its habitat, found mainly in marine water. And when it comes to its body, they are branched, filamentous, and sometimes you observe massive body. So for branched, here you can see laminaria. Just observe the structure. Here this part is frond, this is stipe, and this part is holdfast. Okay, so body is somewhat differentiated here, frond, stipe, and holdfast. This is laminaria, and filamentous forms. Ectocarpus is the example. This is ectocarpus. Then profusely branched like kelp. So this is the profusely branched kelp. Just to observe the structure. Already we studied actually. This algae can reach up to 100 meters. Very massive body. Okay. Now, here pigments. Chlorophyll A and C is present. In Chlorophycean members, chlorophyll A and B is present. In Phaeophyceae, it is chlorophyll A and chlorophyll C. And along with that, you observe xanthophylls and fucoxanthin. The intensity of fucoxanthin makes the algae to have different range of color. Okay. Now, stored food material is complex carbohydrate. Again, about the stored food material I discussed in the previous topic. When the more amount of food is formed, it is stored in the form of different complex carbohydrates. But in Phaeophyceae, please remember this is one distinguishing character in Chlorophyceae, Phaeophyceae, and Rhodophyceae. In Chlorophyceae, it is pyrenoids, but here it is laminarin and mannitol. Laminarin and mannitol is the complex carbohydrate act as a stored food material. Now coming to cell wall, of course cell wall is cellulosic and covered on the outside by gelatinous coating called algin. Now about the algin we already discussed actually. It acts as a hydrocolloid. I told this. These organisms are marine forms. When they are growing in a shallow water, when the low tide condition is present, these organisms are directly exposed to what? It is exposed to sunlight. So at that time, there is a high chance of plant undergoing dehydration. So in order to avoid that, they have a hydrocolloid coating that is called algin. Now, the plant body of Phaeophycean members. Which and all the types are there? Here, little bit here it is differentiated. You can see plant body attached to the substratum by holdfast and has a stalk and stipe and leaf like photosynthetic organs, that is called frond. So holdfast, then stipe and frond, these are the three parts I already shown in the previous picture. Now vegetative reproduction takes place by fragmentation. This is very common. Asexual reproduction in most of the brown algae is by biflagellate zoospores and that are pear shaped. This is very important character. Okay, zoospores are pear shaped and even the gametes are also pear shaped. Okay, so they are biflagellate zoospores, pear shaped and have two unequal laterally attached flagella. This is very important, unequal flagella. That means if I look at the zoospore, Here laterally flagella is present, one is longer and another one is shorter, like this. Laterally attached flagella, one is long, another one is short. Okay, now very important character, please remember. Okay, biflagellate zoospores and unequal laterally attached flagella. Now, sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. Just look at this, within the oogonium. So, this is the character observed in higher algae. Okay, so that is in oogamous species. The gametes are pyriform. Again, it is pear-shaped. Pyriform means pear-shaped. Very important character for your competitive examination. Two laterally attached flagella. That means here laterally attached flagella are of similar sized, but in the case of asexual reproduction zoospores, here also they are pear-shaped, but they are unequal laterally attached flagella. Remember this very important character. Usually in plant kingdom, these asexual spores and in sexual reproduction different types of gametes, questions they take from this part because it is highly confusing. What they want to test actually your ability to understand the difference in the different classification of plants, especially in algae. So the main difference is with the different types of gametes. So please do understand these points are very important for your competitive examination. Now moving on. So these are the some diagrams. Ectocarpus dictyota. See the beauty of this thallus. It is dichotomously branching. Dichotomous branching in the sense, I will show you. See here one structure branched like this, again these are branched, again these are branched, these are branched, these are branched like this. Okay, like this. The goes on, they divide, they branch like this, dichotomously branched. Okay, that is in the case of dictyota. Now next examples are laminaria. See, this picture is very understanding picture because I told about the frond, stipe and the holdfast. Leaf like, stem like and root like. See, this holdfast is attached to the stone. Okay, and here sargassum. These are the air bladders. Here you can see small balloon like structures. They are air bladders. Why they are present? They help the algae to float on the surface. They provide buoyancy. Buoyancy means a floating character. Okay. Then here you can see this is fucus. It is growing on the surface of rock. Fucus. Okay. So these are some examples of Phaeophycean members. I hope the topic related to Phaeophyceae is clear. Please make sure if any point is not understood, let me know in the comment box. Remember the body, then habitat, then pigmentation, and then stored food material, and asexual mode of reproduction where the type of zoospores, how they are present, pear-shaped, laterally attached flagella, and they are unequal. Then sexual mode of reproduction, here also pear-shaped gametes are produced and the laterally attached flagella are of similar sized. Okay, remember all these characters, it is very important. Now all the examples, see, you can remember this easily actually. Now I can tell you: fucus, laminaria, ectocarpus, dictyota and sargassum. See how I can remember this? You can use this technique. FLEDS. This is the shortcut. Fucus laminaria ectocarpus dictyota sargassum. Okay, with the help of this mnemonics, you can remember all the names of the Phaeophycean members. Moving on to Rhodophyceae, commonly known as red algae. Why the color is red? Because they have a dominant pigment, that is R-phycoerythrin. They have chlorophyll A and D. Along with that, they have the R-phycoerythrin. That gives the red color to the body of the plant. So the name Rhodophycean members, commonly known as red algae. Okay, because of R-phycoerythrin. Now, majority marine forms with great concentrations found in the warmer areas. In the sense what? In the region of shallow waters. Okay, but at the same time occurs in both well lighted regions and great depth of the ocean. That means not only found in the shallow regions of the ocean, but also found in the deep down in the water. Okay, so most of them are marine forms, majority of them. There are some exceptions also. There is one algae called Batrachospermum. It is a Rhodophycean member, not given in your textbook. This Batrachospermum, they are found in fresh water. Okay, so multicellular, some of them have complex body organization. Of course, Gracilaria, Gelidium, Polysiphonia, Porphyra, these are the examples. When you look at the thallus structure of these plants, they are complex, multicellular, and some of them are complex body organization. Then food is stored as a Floridian starch, very, very important character again. This Floridian starch, it is similar to the amylopectin and glycogen in structure. In Chlorophyceae, it is pyrenoid. In Phaeophyceae, it is laminarin and mannitol. In Rhodophyceae, it is Floridian starch. It is similar to amylopectin and glycogen in its structure. Okay, so that is about the stored food material. These are very, very important characters. Please do remember this. Okay, these are the examples. Here you can see a person holding Polysiphonia on his hand. Okay, so this is the color of the Polysiphonia, reddish in color. And this is Porphyra, somewhat appears like a plastic wrapper. Okay, this is Porphyra. This is Gracilaria and Gelidium. We studied these two examples where agar is extracted from Gracilaria and Gelidium. Okay, when it comes to the reproduction of Rhodophycean members, there are some very, very important characters here. Of course, vegetative mode of reproduction is by fragmentation. Asexual mode of reproduction is by non-motile spores. Very important character. Have you studied this character in any of the previous classes like Chlorophyceae and Phaeophyceae? No. There we observe zoospores are motile, but here the asexual mode of reproduction is by non-motile spores and sexually by non-motile gametes. See, very unique character. Usually gametes are motile with the flagella, but here again sexually by gametes are non-motile. Then sexual reproduction is oogamous. Only one type. There is no isogamous, anisogamous and oogamous. Here it is oogamous type and accompanied by complex post-fertilization development. Post-fertilization development means when the gametes are produced, smaller male gamete and larger static female gamete, when with the help of water current, when the small male gamete reaches the female gamete and fertilization takes place, nothing but fusion of gametes takes place, then immediately it will not give rise to a haploid plant body. Instead, some post-fertilization changes are observed in this algae Rhodophycean member. There is a reason for this. These Rhodophycean members are the most advanced type of algae in algal classification. So there should be a connection link between algae and bryophytes, right? In bryophytes, we observe some post-fertilization changes. So that is what observed in Rhodophycean member here. Okay. Now I wanted to take one important part of this algae. Let us discuss that. I said in algae, gametophyte this is the dominant plant body. So gametophyte is haploid. Okay. Now gametophytes, they produce gametangia. Now this gametangia is a structure in which gametes are produced. It produces the gametes. Remember, gametes are produced by the process of mitosis. So during the time of mitosis, chromosome number remains same. So the plant body is haploid, gametes are also haploid. Now when these gametes produced, I can say plus and minus, they fuse at the time of fertilization and form a zygote. Just look at the chromosome number, N number and N number. When they fuse, it form a zygote, it is 2N in number. That means zygote is diploid. There is no plant body of diploid in the case of algae. So zygote, very, very important, immediately undergo meiosis. Okay, meiosis is reductional division and then forms the haploid plant body, that is gametophyte. Okay, so remember one character, this is very, very important. Gametophyte produces gametangia undergo mitosis and produces gametes. Gamete fuse to form a zygote. Zygote is diploid, and diploid zygote immediately undergo meiosis and produces the gametophyte. Sometimes in examination, the question may appear like this: zygotic meiosis is the unique characteristics of algae. Please do remember this. Zygotic meiosis, you never see this zygotic meiosis in any of the higher plants. So zygotic meiosis is the unique feature of algae. So zygote undergo meiosis and produces the haploid structure that grow into a haploid gametophyte. Okay, so this is about the reproduction in Rhodophycean members. Now this is one beautiful slide of Rhodophycean members, how they appear in the deep ocean. Okay, just enjoy this slide. Next one, the comparative study of Chlorophyceae, Phaeophyceae and Rhodophyceae. This is very important. Now the questions are taken from this table. Please observe this table carefully. Now some certain characters are taken here. The common name. So for Chlorophyceae, we call it as green algae, Phaeophyceae, brown algae, Rhodophyceae, red algae. That is the common name. The major pigments present are chlorophyll A and B. In Phaeophyceae, it is chlorophyll A and C and fucoxanthin. In Rhodophyceae, it is chlorophyll A and D and phycoerythrin. Okay, this we already studied. Stored food material is starch, mannitol and laminarin, and here it is Floridian starch. Starch in the form of pyrenoids actually. Okay, then cell wall, here it is cellulose and algin. Cellulose is covered by algin, and here cellulose, pectin and polysulfate esters. Okay, here also we can see carrageen covering the cellulose wall. Okay, then flagella number and position. This is very important part to remember. Okay, in Chlorophyceae, 2 to 8 equal apical flagella is present. 2 to 8 equal apical flagella is present in Chlorophycean members. In Phaeophycean members, 2 unequal lateral flagella is present, and very important, in Rhodophycean members, flagella is absent. Okay, then habitat: freshwater forms, brackish water and salt water. Phaeophycean member: freshwater, brackish water and salt water. Most of them are marine forms. Rhodophycean members are, most of them are again salt water forms. Some of them are freshwater forms are also present. Okay, so this is about the overall comparison between Chlorophyceae, Phaeophyceae and Rhodophyceae. Okay, the next one is Bryophytes. Just look at this slide. The pictures appears to be very unique picture, but it is very common on your compound walls. During the season of monsoon, you observe this organism on the compound walls wherever the moist is present. This is the one of the species of Pioneeria. Here you can see the enormous growth of its gametophytic plant body, and these small stalk-like structures are the sporophytic plant bodies. They are producing the sporangium here to bear the spores. Okay, this is the capsule actually. Anyway, just enjoy this beautiful picture of the bryophyte. Now coming to the general characteristics of bryophytes. Very important. Listen to all the characters properly. Bryophytes are slightly advanced plants compared to algae. Okay, so here they are called amphibians of the plant kingdom. Why the amphibians of the plant kingdom? Because the plant body adapted to survive on land, but they compulsory require water for their sexual mode of reproduction. So it requires land as well as it requires water for sexual mode of reproduction. So here they have given these plants can live on soil but are dependent on water for sexual reproduction. This point is very important because once it is asked in NEET examination also. And they usually occur in damp, humid and shaded localities. Damp soil means usually the water content is more. That means almost the surface of the water is covered by water, and in the case of humid, water content is more, and shaded localities means the higher plants will not allow the light to penetrate and reach the soil, and in that region the humidity is more again. Now the plant body of bryophyte is more differentiated than that of the algae. Of course, here the bryophytes are also thallophytes. Okay, that means plant body is not well differentiated. There is no differentiation of true root, stem and leaves. So its thallus is like prostrate or erect. That means it grow straight. Okay, not completely oppressed to the surface. So you observe the erect thallus and attached to the substratum by unicellular or multicellular rhizoids. Rhizoids in the sense root-like structures. Root-like structure. So this root-like structure, sometimes unicellular, sometimes multicellular. Okay, so bryophytes, they lack true root, stem and leaves. They may possess root-like, leaf-like and stem-like structures. So you cannot say it is a root, stem and leaf. Root-like, stem-like and leaf-like structures are present in the bryophytes. Now coming to other characters, the main plant body of the bryophyte is haploid. Very important character. Okay, main plant body is haploid. In the sense, main plant body is gametophyte. This gametophyte is haploid. So it produces gametes. Of course, gametophyte name is because it produces gametes. Hence is called gametophyte. Sex organs in bryophytes are multicellular. This is very important. That means somewhat advanced type of sex organs are produced, and that is called antheridium and archegonium. Here this picture is given just to make you sure that how the sexually reproducing structures are produced. These are archegonia and this is the antheridium, inside which antherozoids are present, and here inside it produces the egg. Okay, so female sex organ is called archegonium and is flask shaped and produces the single egg. This is important. Okay, now the antherozoids are released into the water, where it come in contact with the archegonium, means mouth of the female reproductive structure, and antherozoids fuses with the egg to produce the zygote. So this is the process how the male gamete reaches the female gamete with the help of water. Antheridium produces the antherozoids. Antherozoids with the help of water current reaches the mouth of the female reproductive structure called archegonium, and inside the archegonium single egg is present, so it reaches the egg and fertilization takes place. Now once the fertilization takes place, two haploid gametes, when they fuse, they form a diploid zygote. Now observe this is very important. Zygote do not undergo reduction division. Why they mentioned this? Because previously in algae, zygote immediately undergoes meiosis and produces the haploid gamete. But here it is not like that. Zygote do not undergo reduction division immediately. They produce a multicellular body called sporophyte. See the difference. The sporophytic plant body is not present in algae. Actually, what we consider as a sporophyte in algae, zygote itself is considered as a sporophyte, but here it is not like that. Gametophytic plant body and sporophytic plant body are different. You can differentiate that. But there is one important thing here. They both are not independent. Gametophyte is autotrophic. It can synthesize its own food. But sporophyte is completely dependent on the gametophyte. That means it grows on the gametophytic plant body. Here you can see sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from the gametophyte. Okay, so some cells of the sporophyte undergo reduction division. See, somebody if ask you what is the ploidy of sporophyte, whether you say haploid or diploid, of course sporophyte develops from the zygote. Zygote is diploid, so sporophytic plant body is also diploid. Now sporophytic plant body, just observe here, this is Funaria. This part is the gametophyte. This part is sporophyte. Now some of the cells of the sporophyte inside the capsule undergo meiosis and produces the spores. Remember, spores are haploid. That means diploid plant body of the sporophyte has to undergo meiosis and then produces the haploid spores, right? Now once these haploid spores are produced, now then it is detached from the capsule and fall on the surface of the soil, and haploid spores germinate and give rise to a haploid gametophyte. This is important. Okay, just observe here. some cells of the sporophyte undergo reduction division to produce the haploid spores, and these spores germinate to produce the gametophyte. Now, just observe these pictures. This is the Markantia, one of the example for bryophyte. Just observe the thallus. See, it's somewhat flattened to the surface. And observe the thallus here. One is producing archegonia, another one is producing antheridium. Okay, archegonium is the female reproductive structure, antheridium is the male reproductive structure. One produces the egg, another produces the antherozoite, it is the male gamete. Here one more important structure is given, that is gemma cups. I will discuss about that later in the liverworts, that is the classification of bryophytes. This is asexually reproducing structure on the thallus. When it gets detached, grow into a new plant. It is Markantia. Rhizoids, root-like structure helps the bryophyte plant body to attach to the substratum. Then Funaria, this is a structure, this is the gametophytic plant body, and upper part is the sporophytic plant body. And this is the capsule in which it undergoes meiosis and produces the spores. Now this is sphagnum moss, very commonly observed in the tropical forest. On the surface of the bark you observe this sphagnum moss. Okay, these are some examples. Now coming to economic importance of bryophytes. See, bryophytes, they don't have much economic importance. Some bryophytes are used in medicine, and some bryophytes like sphagnum, they are used as a peat moss. Peat moss in the sense, when these organisms die during the rainy season, they go and compile at one particular place, and they form a black color substances. These black color substances can be used as a fuel, so it is called peat moss. Okay, then very important, they have great ecological importance. Why? Because mosses with the lichens, they establish on the bare rock and they form a sedimentation on it. That means the rock undergoes sedimentation and form a soil, so that in the process of succession, the succession in the sense, the primary bare place will be occupied by the different levels of plants like algae, bryophytes, pteridophytes, gymnosperms, angiosperms, like that. In that process they play a very important role, and sometimes it can be used in packaging of the materials. In olden days, when the thermocols are not there, for the transportation of very delicate materials like glass, they use the these moss. At the same time, if you are transporting the plants from one place to another place, you need to retain the moisture in the soil, so they use the moss to cover the surface of the soil, so it retains water for certain duration of time. That is the economic importance, and they play an important role in plant succession on the bare rocks. This already I explained. Now we discussed about the general characteristics of bryophytes. Now there are classifications in bryophytes. One is liverworts, another one is mosses. Initially we studied two examples. One is Markantia, another one is Funaria. Markantia belongs to liverworts, Funaria belongs to mosses, and even sphagnum moss is there, polytrichum is there, all these belong to mosses. Okay, so let us go with the general characteristics of the class mosses. See, whenever it comes to general character, we are not studying one species character, but in general there are so many species in that particular class, and their general characters are discussed here. Just observe, the habitat is given. Where do we observe the liverworts? Usually in the moist areas, on the moist surface of the soil or moist surface of the bark of the tree, and along with that Here you can see on damp soil. Then marshy ground. Marshy ground means sometimes in some areas you observe water-lodged condition, means water will be retained on the surface, not completely absorbed. Okay, such area we call it as marshy areas. Okay, then deep in the woods means the possibility of penetration of sunlight is very less. So retention of moisture will be very high, humid condition will be there. In such area, it is the favorable condition for a growth of liverworts. So we observe in the deep woods. Now, plant body of liverworts is thalloid. Of course, in general characters of bryophytes, we already studied this. Thalloid means plant body is not completely differentiated. Okay. Example is Markantia. Now, this character is interesting. The thalloid body is dorsoventrally differentiated. That means you can clearly differentiate dorsal side and the ventral side. Okay, there are some characters like having trichomes or rough surface, smooth surface, like that. So that character help the thallus to differentiate its dorsal and ventral surface, and it is closely oppressed to the surface of the soil. In the sense, it is closely adhered to the surface of the soil. Now, the leafy members have tiny leaf-like appendages in rows on the stem-like structure. Here, leaf-like structure, stem-like structure. That means it is not a true leaf and true stem. So stem-like structure, leaf-like structure. Here you can see the Markantia thallus. So this appears to be a leaf-like structure. Okay. Now, next one, asexual reproduction. Very common type in all the algae. Asexually they reproduce by fragmentation. Other than that, there is one special character here. Just observe, gemma cups. In plural it is called gemmae. Here you can see this is one gemma, this is one gemma, here one more, like this. Okay, they are gemma. Now they are present inside the gemma cups. So what is the role of this structure? Just like a buds. Okay, when the thallus on onset of unfavorable condition, when thallus dies, this gemma caps, they detach from the thallus and they grow into a new plants. So this is one type of asexual mode of reproduction observed in liverworts. Now, there was one question appeared in NEET based on the same topic. I decided to show this question on this slide so that you will have certain idea about that. In bryophytes, the gemma helps in which of the following? Options given are nutrient absorption, gaseous exchange, sexual reproduction, asexual reproduction. Very simple and direct question. The answer is asexual reproduction. Next one. Okay. So here, this is the characteristics of gemma cup. Okay, gemma. Gemma are green. All the characters are important because once it was asked in NEET. It is green in color, multicellular, and asexual buds which develop in small receptacles called gemma cups located on the thalli. This entire statement you should remember. So what is the character? Multicellular, green, asexually reproducing buds, and which develop on the gemma cups. Okay, so the gemmae become detached from the parent body and germinate to form a new individual. This I already explained. Now, during sexual reproduction, male and female gametes are produced inside the gametangium. Gametangium is a multicellular structure. That we call it as antheridium and archegonium. Now, those gametes, they are antherozoids and egg. With the help of water current, the antherozoids reach the egg and fertilization takes place. This is all common phenomena. And zygote formation takes place. Now this is very, very important. Bryophytes, we already studied that sporophyte doesn't have independent life. So the zygote develops on the gametophyte and produces the sporophyte. So sporophyte is differentiated into foot, seta and capsule. Okay, foot, seta and capsule. So this diagram here given is not liverwort. Just to make you understand foot, seta and capsule, I just taken this diagram. Otherwise this belongs to mosses, not liverworts. Okay, so foot, seta, capsule, where foot is embedded in the gametophyte, which helps in absorption of nourishment from the gametophyte, and seta is the connective part to the capsule, and inside the capsule certain cells undergo meiosis and forms the spores. Remember this, zygote is diploid. Sporophytes, they develop from the zygote. So sporophytic plant body is diploid. Now the cells inside the capsule is initially diploid only. To produce the haploid spores, they should undergo meiosis. So spores produced are haploid, and those spores, they give rise to haploid gametophyte, and gametophytes are free living and they are independent and they are autotrophic. Okay, so that is about the liverworts. Now there is one more question which was already asked in your NEET examination. The correct sequence of events in the life cycle of bryophyte is? Answer I already given here. Just look at the pattern of the question. Slowly from 2024 to 2025, they are focusing on the lengthy questions. So then only the students will learn to manage their time. Only the one who knows the concept very well, they can go with the speed. Others, they cannot, even though they know the concept. Now just observe this: fusion of antherozoids, attachment of gametophyte to the substratum, reduction, division to produce the haploid spores, formation of sporophyte, release of antherozoid into water. There is a sequence here. Just attachment of gametophyte to substratum. This is the first one. Then release of antherozoids into water. Then fusion of antherozoids with the egg, and then formation of sporophyte and reduction, division to produce the haploid spores. So this is the sequence of life cycle of a bryophyte. Okay, next, moving on, mosses. Not very common, but in rainy season you can see this mosses. Where is the common place we observe this? In case in trekking time or any other forest visit, if it is a tropical forest, then moisture is very high. At that time, on the tree trunks, we commonly observe this mosses. Other than that, in rainy season, if the compound wall is moist or interlocks are moist or the surface of the soil is moist, and at that time you can observe the funaria that also belongs to mosses. Now, predominant stage of the life cycle of the moss is gametophyte. Of course, not only in mosses, the entire bryophytes, predominant stage is gametophyte only, and which consists of two stages. Here there is a reason to write this predominant stage. The reason is there is a peculiar character in gametophyte. Whatever we studied in Markantia, there is no much differentiation in gametophytic generation. But here in gametophyte we observe protonema stage. See, here I purposefully included one diagram which was not given in your NCERT. Look at this. This is the spore. Okay, spore is haploid because it is produced inside the sporangium, and destiny of the spore is to germinate and produce a gametophyte. If gametophyte is haploid, then spore should be haploid. Okay, so this spore, when it germinate, yes, here, when it germinate, it produces a filamentous like structure that is called protonema stage. Okay, now this structure, protonema structure, then start to produce leaf-like appendages on it. It is not leaf, leaf-like structures. So we call it as leafy stage. Okay, or in other words, it is secondary protonema. And then they start to produce the lateral buds. So here you can see the lateral buds. Okay, so this is about the protonema stage, a very important gametophytic lifespan which is observed in the mosses. Now, moving on to next one. There was one question in NEET. It is assertion reason type. The first stage of gametophyte in the life cycle of the moss is the protonema stage. Reason: the protonema develops directly from the spores produced in the capsule. Answer I have already given here. Both assertion and reason are correct, and R is the correct explanation of A. Because it is a sequence. The first stage of gametophyte is of course protonema. In case if they have mentioned here the first stage of gametophyte is secondary protonema or leafy stage, then the assertion is wrong. Okay. Just observe the pattern, how they are choosing the questions. Okay. Then continue with the topic. Secondary protonema consists of upright, slender axis bearing spirally arranged leaf-like structure. It is not leaf. Okay. I have already shown you a picture how the leaves are arranged. If you want, you can check one more time here. Just observe, leaf-like structures are arranged on the stem-like structure. Okay. Now they are attached to the soil through the multicellular and branched rhizoids. Just observe here. Just these are the rhizoids, branched, multicellular, and they are branched. Now this stage bears the sex organs. Sex organ is antheridium and archegonium, because whatever we are observing, that protonema is a gametophyte. Gametophyte should produce the gametangia. Gametangia is antheridium and archegonium. They bears the gametes. Gametes are haploid or diploid? Of course they are haploid, because gametophyte is haploid. Mitosis takes place inside the antheridium and archegonium, which leads to the formation of male and female haploid gametes. Okay. Now vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. Please do remember this word, budding in the secondary protonema. Just like fragmentation, but a small buds arises from that, and that will detach and give rise to a new plant. Now coming to sexual mode of reproduction, very important topic. Here I have mentioned so many questions appear to the NEET from this topic. Sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoot. If you want, you can observe this here. See here, yes, this structure here you can see, this structure, this is archegonium and this is antheridium structure. This is antheridium. These are the antherozoids. And here cup-like structure, that is the archegonium. Okay. Now, after fertilization, after once antherozoids are released, with the help of water current, it reaches the mouth of the archegonium or neck of the archegonium, then enters, then fertilizes with the egg, and it forms the zygote. Now, zygote develops into sporophyte, very important. Zygote develops into sporophyte consisting of foot, seta and capsule. This already I explained. Foot is embedded in the gametophyte, seta is the upper part, and capsule bears the spores again. And this sporophyte is completely dependent on gametophyte. It is heterotrophic. Okay. Now, sporophyte in mosses is more elaborate than in the liverworts. Elaborate in the sense their life cycle is somewhat duration is more, and the capsule contains spores. Then spores are formed after meiosis. This is important. Why there is a need of meiosis, reductional division? Because sporophyte is diploid. Okay. The mosses have an elaborate mechanism of spore dispersal. See, sometimes we observe this in higher plants like angiosperms, especially impatiens balsam plants. They produce type of seeds inside a capsule, and when it matures, if you just touch it, it blasts and spread the seeds. Similarly, there is one mechanism here in the dispersal of spores. They have a special cells like elater cells. When there is a moisture in the atmosphere, these elaters absorb it and they swell and helps in the burst open of the capsule, and then spores are released. See, these are some pictures you can clearly observe. The first one, just observe this. This is Funaria, very common on the wall of the compound walls. And this is Polytrichum, slightly different from Funaria. And the third one is Sphagnum moss. Sphagnum moss. In tropical forest, you can see the Sphagnum moss. Okay. Students, let us continue with the topic Pteridophytes. See, the name starts with the letter P, but we pronounce it as Pteridophytes. We studied algae, which is the most primitive one, where plant body is not differentiated, and we call it as thallus. Similarly, we studied bryophytes. There also the plant body cannot be differentiable, so it is also known as thalloid. But when it comes to pteridophytes, here you can clearly differentiate root, stem and leaves. The first slide here which I have shown, it is a massive growth of fern in the forest. Okay, this is the floor of the forest. Here you can see the large trees full of ferns. So pteridophytes, they commonly grow in shaded areas. Okay, they also require water for their sexual mode of reproduction. Compulsory they require water for sexual mode of reproduction, because here there is no structures which helps in dispersal of gametes. Okay, so we will study that in detail. Okay, pteridophytes includes horsetail and ferns. So the name is mainly because of its structure. Okay, I will show you the picture of horsetail and ferns. So mainly we study about these species. Now pteridophytes are used for medicinal purpose. See, these are all very little economic importance, because there is no much importance about its ecological importance. Anyway, now sometimes the people who are living in forest areas, they know how to use it as a medicinal plant. So they use the ferns as a medicinal plant, or any of the pteridophytes. In that case, I don't know about that. So they are also frequently grown as ornamentals. Commonly, if you visit any of the what we can say gardens or any of the beautiful places where they grow ferns for ornamental purposes, okay, then evolutionarily they are the first terrestrial plants. This is important. This is very, very important. Rest of the things are just introduction to pteridophytes. Evolutionarily they are first terrestrial plants because bryophytes are called amphibians of the plant kingdom, but next to that pteridophytes, they have a true root and adapted to land environment, okay. They have xylem and phloem as a vascular bundle. So we'll study in detail about the xylem and phloem in anatomy of flowering plants topic, but remember this, xylem is meant for transport of water, phloem is meant for transport of food, okay, that we will remember. Here afterwards, after the pteridophyta, gymnosperm and angiosperm, they all have vascular bundles, that is xylem and phloem. So this is the point where we can say clearly differentiate vascular plants and non-vascular plants. Algae and bryophytes are non-vascular plants, pteridophyte, gymnosperm and angiosperms are the vascular plants. Now, pteridophytes are found in cool, damp, shady places, though some may flourish well in the sandy soil condition. So this is the habitat where we commonly observe the pteridophytes. Okay. Pteridophytes, the main plant body is sporophyte. Now listen carefully, this part is very, very important. We should know what is sporophyte, what is gametophyte, okay. So main plant body is sporophyte. Why we are considering so much about this now? Because in algae the main plant body is gametophyte, bryophyte main plant body is gametophyte, but in the case of pteridophytes main plant body is sporophyte. Then in gymnosperm main plant body is sporophyte, angiosperm main plant body is sporophyte. So this we should know, okay. So dominant plant body is sporophyte. Now the fern what we observe, or any of the examples of pteridophytes, that is the sporophytic plant body. Hope you all seen the ferns. First slide I have given the example of a fern. So that is the sporophyte. Now this sporophytic plant body produces the definite leaf, that is called sporophyll. Phyll is representing the leaf. It is produced on the sporophyte, so the name sporophyll. This sporophyll may be small, that is called microphyll. If it is large, then it is called macrophyll. So here example is given for microphyll. Microphyll in the case of Selaginella and macrophyll in the case of ferns. Okay, so this example please do remember, means if the leaf is small then it is called microphyll, if leaf is large then it is called macrophyll. In ferns leaves are large, pinnately compound leaves, okay. The sporophyte bears sporangia. What is this sporangia actually? Inside which spores are produced. Remember always sporophytes are diploid. Now sporangia is also diploid. Now cells inside the sporangium undergo meiosis and produces the spores. Okay, these spores are haploid. Now spores on dispersal give rise to a haploid gametophyte. Okay, so sporophyte bears sporangia that are subtended by leaf-like appendages called sporophyll. This is very interesting. Here sporangium is produced on the leaf and that leaf is called sporophyll. I will show you the picture how they are arranged. Many sporangia are present on the leaf, that is called sori. Okay. Now, in some case, sporophyll may form a distinct compact structure called strobili or cones. It may not be same in the case of all the species of Pteridophyta. In some Pteridophyta, there will be a central axis like this. Surrounding this, these are, let us consider these are the leaves. Okay. Now these leaves, they bear the sporangia. Okay, and they are forming a cone-like structure. This is the cone-like structure, right? This is not same in the case of all the species. Okay, so this is observed in Selaginella and Equisetum. There is a specific example of pteridophytes. They show cones, or we can say strobili. Don't get confused with this. Now, pteridophytes, the sporangia produces spores by meiosis. This I already explained. The meiosis takes place in spore mother cell. The cell which give rise to spores by meiosis are called spore mother cells. The spores germinate to give rise to this statement is very, very important. Inconspicuous means very small. Okay, cannot be observed through naked eyes. Small but multicellular structure, free living, mostly photosynthetic. This term is also important. Mostly photosynthetic in the sense, if there is no proper sunlight reaching the gametophytic plant body, at that time sometimes it acts as a heterotroph. Okay, so that is why here it has given mostly photosynthetic. Thalloid gametophytes called prothallus. So this is the technical term. Okay, technical term used in okay. So prothallus is the technical term used for gametophyte. See, just observe here, observe this diagram. It's a photography of prothallus. It is somewhat heart shaped. Okay, it is a multicellular structure, inconspicuous. Okay, you should remember all these terminologies: inconspicuous, small but multicellular. They are free living, not dependent on the sporophyte, and mostly photosynthetic, thalloid gametophytes, and its name is prothallus. One of the very important concept for NEET. Please do remember this. Okay. Next one, pteridophytes. In that continuation part, the gametophytes require cool, damp and shady places to grow because of this specific restricted requirement and the need for water for the fertilization. The distribution of pteridophytes are restricted to some geographical area. So this is very important. Pteridophytes are not common just like angiosperms, right? Sometimes we rarely observe these pteridophytes. What may be the reason? Because they need damp soil, cold climatic conditions, and they require compulsory water for their sexual mode of reproduction. So these are the limitations. Because of these limitations, you cannot see everywhere the growth of pteridophytes. Only in some area we observe the pteridophytes. So their distribution is restricted to narrow geographical area. Okay, so the gametophytes bear male and female sex organs called antheridia and archegonium respectively. Here again I have shown one more picture of the prothallus. Don't think the prothallus is this much big. It's very small. It is just a model. Okay, model of the prothallus. It is not real structure. Here you can see a heart-shaped multicellular structure of prothallus, and on that you can see the rhizoid-like structures, root-like structures, helps the gametophyte to adhere to the surface. And here you can see archegonium and antheridium. Inside that, egg and antherozoites are produced. See, the produced antherozoites, they require water to reach the mouth of the archegonium, then only the antheridium reaches the archegonium, and inside that it fuses with the egg and fertilization takes place. Once the fertilization takes place, what happens? Zygote formation takes place. Zygote is diploid because it is formed by the fusion of two haploid gametes. Now diploid zygote, now what happens to this diploid zygote? Of course, there is a sporophytic generation, so it has to form a sporophytic plant body. But there is one important character here, very important character. This zygote start to form a sporophytic plant body, but for a certain interval of time, that sporophytic plant body retains inside the female gametophyte. Very important, okay? Sporophytic plant body retains inside the female gametophyte, and this is the precursor for seed habit. Precursor means what? Starting point, from where the seed habit will start. Okay, you can, if you observe the higher plants like Gymnosperm and Angiosperm, so what is the sporophyte actually? The embryo which is present in the seed then grow into a sporophyte. That embryo retains inside the female gametophyte, right, for a certain interval of time. So same character is observed here also. So that is called precursor for seed habit. Okay, here one question is asked in NEET. Which one of the following is false? Gametophytes are multicellular, tiny and photosynthetic thallus. This statement is correct. Prothallus is another name for sporophyte. This statement is wrong. We know that prothallus is another name for gametophyte. Gametophyte is the predominant phase in mosses. That is correct. Because in algae and bryophytes, moss comes under bryophytes. Gametophyte is the dominant plant body. But afterwards, in Pteridophytes, Gymnosperm, Angiosperm, sporophyte is the dominant plant body. Strobili, a distinctive cone-like structure, is present in Selaginella and Equisetum. Last slide, we studied this. Okay. Next. This already I explained, but let us see what is given on the slide. Water is required for transfer of antherozoites. The male gamete released from the antheridia to the mouth of the archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of a zygote, and zygote thereafter produces multicellular well differentiated sporophyte, which is dominant phase of the pteridophyte. So this is a simple mechanism how the antherozoites reaches the mouth of the archegonia and then reaches the egg, and how the fertilization takes place. After fertilization, zygote is formed and zygote develops into sporophyte, and sporophyte is the dominant phase of life in the case of pteridophytes. Now, so this is very important again. In majority of the pteridophytes, all the spores are similar kinds. Again, this point here is very important for your competitive exam. Okay, heterosporic condition. What is this heterosporic condition? Homosporic condition and heterosporic condition. Suppose produced spores are of all same sized, then it is called homosporous condition. If the produced two spores are of different sized, then it is called heterosporous condition. So what happens when heterosporous condition is present? So it is observed in plant like Selaginella and Salvinia. Okay, in Selaginella and Salvinia, we observe the heterosporous condition. Okay. Now, when this heterospore germinate and produce the gametophyte, in that microspore produces the male gamete and megaspore give rise to female gamete. Okay. in the sense gametophyte, gametophyte. Microspore give rise to male gametophyte, megaspore give rise to female gametophyte. This is important respectively. So the female gametophytes in these plants are retained on the parent sporophyte for variable period. This is what I explained. It is the precursor for seed habit. If you want, I will repeat that one more time here on this slide. See, the development of the zygote into young embryo takes place within the female gametophyte, and this event is a precursor to seed habit. Precursor means starting point. Okay, just observe here. Now, microspore mother cells undergo meiosis and producing the microspores. At the same time, this is the sporophytic plant body. On the sporophytic plant body, it produces the spores. On the gametophytic plant body, that is the prothallus. So here is the prothallus. This prothallus produces the antheridium and archegonium. Antheridium produces antherozoites. Archegonium produces egg. Okay, now antheridium having antherozoites with the help of water current reaches the mouth of the archegonia and then fuses with the egg. Fertilization takes place, forms the zygote. Now zygote develops into sporophyte. Now this sporophyte is not immediately detached from the archegonium. Instead, for some interval of time, it retains inside the archegonium, and that we call it as precursor for seed habit. So this event is very important in the point of evolution, because so far what we observed where there is no seeds, but the next classification, gymnosperm and angiosperm, they show seed habit. So this is the starting point for the seed habit. Now these are some of the example. Please just observe all these example, very nice pictures. This is xylotum. The plant species is xylotum, belongs to the class Xylopsida. Xylopsida. There are 4 classifications: Xylopsida, Lycopsida, Spinopsida and Pteropsida. So the first Xylopsida containing one plant species, that is xylotum. In your textbook they have given one example. And the second one, which comes under Lycopsida, this is Selaginella, very common on wet walls. This is Selaginella. Lycopodium, just observe the cones here. We studied it, strobili or cones. The microsporophylls or sporophylls bearing sporangium, they all together forms the cones or strobili, and that's what we studied, right? This is Equisetum. Now this picture you just observe carefully, it's very common fern. Now this is the dorsal surface of the leaf, nothing but the sporophyll. When the leaf matures, in its dorsal surface, it produces many sporangium. Each one is a sporangium here, and we call it as sori. Inside that, it produces numerous spores. Okay. This is one more example, Teres. And this is the backward photocopy of this plant. When plant matures, when its leaf matures, this is the sporophyll bearing sporangium on its margin, that we call it as sori. Inside that, numerous spores are produced. Now this is called adiantum, commonly known as walking fern. Why the name walking fern? The tip of the leaf, when it touches the ground, it starts to produce the root and produces another plant. So this is one of the mode of vegetative reproduction observed in adiantum. Okay, so this is one leaf and this is the plant. Students, let us continue with the topic, gymnosperms. See the name, gymnos. Gymnos means naked. Sperms meaning seeds, naked seeded plants. Just observe the first slide, a beautiful slide of gymnosperm, where one cycas plant is bearing the cone here. So this is the cone, structure of a cone. Observe this properly, very nice slide. Next one, we'll go with the general characteristics of gymnosperms. I already told what is gymnos meaning naked, sperma means seeds, naked seeded plants. Now, seeds that develop post fertilization are not covered and they are naked. That means here there is no fruit, seeds are directly present. Gymnosperms include medium sized trees to tall trees and shrubs. Let me give you some examples. Cycas, hope you have seen in some gardens. Then pinus, very common pine trees are present in mountains like Himalayan regions. Then ginkgo, it's very rare plant, but it has evolutionary significance. Then Sikova, a very large giant tree. I will show you the picture of that. Okay, so these are some of the examples of gymnosperms. Okay, and ginkgo is also known as a living fossil because very few number of plants are available, so they are called living fossils. There is one question asked in NEET, very simple question, nothing to worry about. Which one of the following is the characteristic feature of gymnosperm? Here 4 options given. Seeds are absent, gymnosperms have flowers for reproduction, seeds are enclosed in fruits, and seeds are naked. Of course, the seeds are naked is the correct answer. Now coming to the character. Now initial characters about gymnosperms are very important. Some of the adaptations are given. In gymnosperms, as we know, they are well advanced plants because pteridophyte itself, they have well differentiated plant body, that is true root, stem and leaves. In gymnosperms, here we observe taproot system. Taproot system means one mother root, then primary root, then secondary root, then tertiary root, root hairs, they are all present in taproot system. Now, there are some adaptation in this taproot system in some members of the gymnosperms. In Pinus, mycorrhizal association is observed. Okay, this mycorrhizal association helps in such a way that mycorrhizae is the fungus. Then plants are autotrophs, have the ability to synthesize their own food. So this heterotrophic fungus depends on the autotrophic plant. In return, mycorrhizae absorbs water from the greater depth of the soil and give it back to the plants. So here both the organisms are mutually benefited. So one type of symbiotic association is observed between the mycorrhizal fungus and the roots of the pinus tree. Now coming to the next adaptation in the case of cycas. If you observe the stem of the cycas, you observe pinkish color coralloid roots. These coralloid roots, they have N2 fixing cyanobacteria. Okay, inside that cyanobacteria is present and they have the ability to fix the atmospheric nitrogen. Okay, that is one more important character. Then coming to the stem of the gymnosperms, in case of cycas, stem is not branched, unbranched in the case of cycas, but in pinus it is branched. Okay, then leaves, in some plant we observe simple leaves, in some plants it is compound leaves. When it comes to the leaves of cycas plant, it is pinnately compound leaf, very large, and not very large, large leaves, and can persist up to few years. Okay, so that is one of the adaptation. Then in the case of pinus, leaves are very small, it is like needles, needle like leaf. That means surface of the leaf is reduced to a very small area. so that they can avoid the loss of water from the aerial part of the plant. And they have a special type of stomata, sunken stomata. That means stomata is not present on the epidermis, it is present just below the epidermis. Okay, that condition is called sunken stomata. This is also an adaptation to reduce the loss of water from the aerial part of the plant. And they have thick cuticle, that is one more adaptation to avoid the loss of water from the aerial part. You know what is cuticle? It is a waxy layer. If thick cuticle is present, then loss of water will be reduced. Leaves in Gymnosperms are well adapted to withstand extreme temperature, humidity and wind. That is the adaptation, because you know where they grow. Actually, pinus tree, they grow in very harsh condition. Gymnosperms, in that we were discussing about the spores, different types of spores. Even some of the members of Pteridophytes, they showed heterosporous condition. Likewise, in Gymnosperms also we observe heterosporous condition. Heterospory means now produced spores are of not same sized. One is small, another one is large. That condition we call it heterosporous condition. Now here, two kinds of spores are produced within the sporangia. Sporangia means it is a structure inside which spores are produced. Now, born on the sporophyll. Now, phyll means leaf. Sporophyll means spore bearing leaf. Now, this sporangia are produced on the spore bearing leaf. Okay, now these leaves, they all arrange on the one axis and form a compact strobili, or we can say cones. Okay, so which are arranged spirally along an axis to form a lax or compact strobili or cone. So this is the description of arrangement of leaves, sporophylls, especially sporophylls, spore bearing leaves. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. See, there is nothing to confuse, just remember. Let me take one leaf as an example. So this is one leaf. Okay, suppose now this leaf is bearing sporangium, then the name of this leaf is microsporophyll. Okay, this microsporophyll having microsporangium, this is the microsporangium. Now tell me, sporophytes are haploid or diploid? Sporophytes are diploid. That means microsporophyll is diploid. Now the cells inside the microsporangium, this is called microspore mother cells. They undergo meiosis and then produces the spores. So these are nothing but the microspores, or in other words we can say pollen. Okay, so how this microsporophyll is arranged? Actually, there is one central axis like this. Surrounding this, these microsporophylls are arranged like it appears like a cone. All these are the leaves. See, so how they are arranged? Here you can see arranged spirally along an axis. So this is the axis and spirally they are arranged, and this we call it as male cone. Similarly, the female cone is also formed where it has megasporophyll. On the megasporophyll, megasporangium is present. Inside the megasporangium, megaspore mother cell, nothing but the sum of the nucellar cells, they are undergoing mitotic division and form a megaspore mother cell. Now megaspore mother cell undergo meiosis and form 4 haploid cells. Out of the 4 haploid cells, 3 cells they degenerate and remaining 1 cell that forms the female gametophyte. Now there is one important character here. Now this is the comparison with angiosperms and gymnosperm. In angiosperm, in higher class you will study, it is monosporic development. One female gametophyte produces 1 egg. But in the case of gymnosperm, one female gametophyte produces more than 1 egg. Okay, so more than 1 archegonia are present here. So this reduced gametophyte is called pollen grain. That means in this gymnosperms, there is no independent gametophytic lifespan. See, if we compare this gymnosperm with the lower plants, algae has independent gametophytic generation, Pteridophytes has independent gametophytic generation, then Bryophytes has independent gametophytic generation. But when it comes to gymnosperms, sporophytic generation is the dominant, and gametophytes, they don't have independent existence. They have to dependent on the sporophytes. In the sense, here the gametophytic lifespan is very much small. What we can say, pollen grain itself is a gametophyte. It is very short interval of time. Okay, so the development of the pollen grain takes place within the microsporangium. Now don't get confused, it's very important part of the topic. What is microsporophyll? Leaf which bears the microsporangium. What is microsporangium? Structure in which pollen grains are produced. Now what is pollen grain? Actually a gametophytic lifespan. Now pollen grain, when it is present inside the microsporangium, then that leaf is called microsporophyll. Now arrangement of many microsporophylls forms the male cone. Okay, hope this concept is clear. Now here you can see the picture also. In the case of megasporophyll, what I explained actually, megasporophyll, a specialized leaf which bears megasporangium. Here you can see the megasporangium. This ball-like structure is actually a ovule. It is not a fruit. Okay, gymnosperms, they don't produce fruits. Seeds are directly exposed. So this is the ovule actually. Now there is no ovary here. Megasporophyll, these structures are megasporophylls. Here you can see this is the megasporophyll. Okay, this megasporophyll bears the megasporangium. Now this is the megasporangium. Here you can see these are all the megasporangium. Inside the megasporangium, what happens? Some of the cells called nucellar cells, they undergo mitosis and forms the megaspore mother cell. Now megaspore mother cell undergo meiosis because gametophytic lifespan is haploid, right? So megasporangium being diploid has to undergo meiosis and produce the haploid megaspores. Now nucellar cells undergo mitotic division and forms the megaspore mother cell. Now megaspore mother cell undergo meiosis and forms the 4 haploid cells. Now in that 4 haploid cells, 3 cells undergo degeneration and 1 cell retains. Now this cell develops into female gametophyte. Remember this, it is called female gametophyte. Now female gametophyte bears the 2 or more archegonia, nothing but 2 or more eggs are present in this gymnosperms. This is the unique character which is not observed in angiosperms. In angiosperm it is monosporic development, very important character. Okay. So the male and female cones or strobili may be born on the same tree like pinus. Here you can see this is the male cone and this is the female cone produced on the same branch. So what is this condition actually? Monoecious condition. Monoecious condition, remember this term. Monoecious condition. Now here you can see both are cycas plant. One plant is bearing a male cone. Here another plant is bearing female cone. Now for this condition we call it as dioecious. Monoecious condition and dioecious condition. Okay. In cycas, male cones and megasporophyll are born on the different trees and that we call it as dioecious condition. Now moving on to next. Megaspore mother cell differentiate, this I already explained actually how the megaspore formation takes place. Just I will read out. The megaspore mother cell is differentiated to form one of the cells of the nucellus, and nucellus is protected by envelopes and the composite structure called ovule. The ovules are born on the megasporophyll which may be clustered to form a female cone. Then megaspore mother cell divide meiotically to form a 4 megaspores. One of the megaspore enclosed within the megasporangium develops into multicellular female gametophyte and that bears 2 or more archegonia or female sex organs. 2 or more archegonia. This is important actually. Okay, the multicellular female gametophyte is retained within the megasporangium. Unlike bryophytes and pteridophytes, gymnosperms, the male and female gametophytes do not have independent free living existence. The remaining remain within the sporangia retained on the sporophyte. I just draw some of the diagrams so that you can understand this properly. Let us take one sporophyll. So this is the sporophyll. Okay. Now this sporophyll is bearing the sporangium. So let us consider this is the, here I will label megasporophyll. This is the megasporangium. Now we are focusing on this megasporangium. Let us take megasporangium here. Now this megasporangium having cells inside, this is diploid, right? So it has nucellar cells. Nucellus. Okay, this nucellar cells, they undergo mitotic division and form a megaspore mother cell. Now, one of this megaspore mother cell, now here I will take, this is the megaspore mother cell, whether it is diploid or haploid. Again, ploid is very important. Now this is being diploid, undergo meiosis. Now when it undergo meiosis, what happens? I am writing diagram here. When it undergo meiosis, again it forms the 4 haploid cells. Okay, out of this 4 haploid cells, 3 cells they degenerate, and the cell which is near to the micropylar region, will be retained. Now we discuss about this micropylar region cell, that one haploid cell. Now this is the haploid cell. This cell form a female gametophyte. Now this female gametophyte forms the two archegonia. Okay, so this is how the egg formation takes place inside the megasporangium. This is what here they explained. Now this egg is haploid or diploid? Of course it is haploid. Okay. Now here there is no ovary wall, so there is no fruit formation. Ovule directly develop into seed. The pollen grain is released from the microsporangium. This is the process. Just like what we observe in angiosperms, when the flowers open, pollen grains either it is carried by the insect, or it is carried by the wind, or it is carried by the water, or it is carried by the animals. Finally, it reaches the stigma of the female flower. Similarly, that we call it as pollination. Here also pollination takes place. Male cones having microsporangium produces numerous pollen grains. These pollen grains are carried through the wind and reaches the mouth of the megasporangium, where the pollen grains they germinate and produces pollen tube, and the pollen tube carrying male gamete reaches the micropylar region, and in the micropylar region it fuses with the egg and form a fertilization. Now fertilization gives rise to zygote formation. Now zygote develops into embryo and ovule develop into seeds, and there is no ovary, so seeds are directly exposed, so the name Gymnosperms. Okay, so that is what given here, the total the event which occurred during the pollination process. So these are some of the examples, very important. Giant Sicova. Hope you can see the people, they are standing in front of this tree. Look at the size of the tree, very giant tree, name Sicova. And this is Cedrus. Look at the cones, slightly different compared to Pinus. Okay, Cedrus plant. And this is Ginkgo biloba. This has some evolutionary significance. It is commonly known as living fossil. Okay, Ginkgo biloba. The fruits produced by these plants have some bad smell. And this is the Pinus cone, male cone, and the female cone. Cycas plant. In cycas, it is dioecious condition. Male plants and female plants are different. Okay. So last part of the topic plant kingdom, that is angiosperms. Without this topic will not be completed. But about the angiosperms, the description given is not much, because in the second PUC we study in detail about the sexual reproduction in flowering plants. In that they have covered almost all the things related to angiosperms. So in the first year topic they have just given a brief account of this angiosperms. Now in angiosperms, these are the flowering plants. So production of flower is the distinguishing character of angiosperms, and they have well developed vascular system, plant body is differentiated into true root and stem. These are all very important characters. Here two examples are given. The smallest angiosperm is Wolffia. It's very small. You can hold the hundreds of plants in one hand. And eucalyptus is the largest tree, which may grow up to 100 meters. Okay. Then here we can classify them as monocotyledons and dicotyledons based on the number of cotyledon in the seeds. Rest of the things we will discuss in the higher classes. So this is the picture of dicot plant. This is the picture of monocot plant. There are so many differences. Here it is taproot system, here it is fibrous root system. That all we study in anatomy of flowering plants. Even in morphology also we study the difference between monocots and dicots. Okay. So with this we completed the topic. Let us see some of the questions related to this. So this is the question asked in NEET. Pteridophytes, Bryophytes, Angiosperms and Gymnosperms. You should match the list 1 with the list 2. Salvia, Ginkgo, Polytrichum and Salvinia. When match the following is given, if we know 1 or 2 examples, then it is easy to solve the problem. We know Ginkgo belongs to Gymnosperm, just now we studied. And here Polytrichum, Salvinia belongs to which one? Salvinia belongs to Pteridophytes and Polytrichum belongs to Bryophytes. Of course, then Angiosperm it is Salvia. So this is how we'll match the questions. Then which one of the following is not found in Gymnosperms? Of course, very easy question. That is flower. Flower is not found in the Gymnosperms. Yes. So with this we completed the topic plant kingdom.